All right, welcome everyone. Let me introduce Dr. Haibo Liu. He just started new job at UMass Medical School, and he will talk about best practices for ATAC-seq and quality control and data analysis. So Haibo, please go ahead.
Good morning, everyone. So in my workshop, I can talk about the best practice for ATAC-seq data QC and analysis. So at first, I would like to thank my collaborators. So Jianhong from Duke University, who is a major developer of the ATAC-seq QC package, and the junior is our supervisor, Jianhong's previous supervisor, and the other collaborator from the UMass Medical School. And also thank the BIOS Conductor Conference team for organizing this meeting. Before I start the QC, ATAC-seq data QC and analysis, I would like to give a brief introduction to the ATAC-seq technology. So in eukaryotic cells, the nuclear DNA are packed into chromatin. Different chromatin regions are packed to different level, with some region which is tightly packed, which is called closed chromatin, while some other region which are nucleosome depleted. So those regions are accessible to the transcription machinery and the probing molecules such as the DNase1 or the TM5 transposase. Those regions are called open chromatin. Even though the percentage of open chromatin is low, less than 5% across the genome, but more than 90% of the transcription factor binding sites are located in the open chromatin region. So it's very important to identify the open chromatin region to understand the gene regulation. Over the years, quite a few methods have been developed to profile the chromatin accessibility, such as mNA-seq, DNA-seq, the FAIR-seq, and more recently, the ATAC-seq. Compared to the other methods, the ATAC-seq has several advantages, such as it only requires 500 to 50K cells, and also have a very short protocol. It can be finished in a few hours. So this method getting popular and widely used in many different fields. The wet lab protocol for ATAC-seq assay is pretty simple. So it's composed of majorly two steps. First, use the preloaded TM5 transposase to cut and append the sequencing adapter to the chromatin fragments. Then the PCR was used to amplify the fragments. By PCR process, 128 bases of adapter sequence appended to the ends of the genomic DNA fragments. So this technology has been used to profile the open chromatin regions and also infer the nucleosome positioning and identify the transcription factor footprints and also infer the gene regulatory network. More recently, in combination with the single cell technology, ATAC-seq have been used to determine the cell state and identity of identities. So even though the wet lab protocol for ATAC-seq assay is pretty simple, so we still need quite a strict QC steps during the process, including the nuclei QC, so we can check the nuclei quality under the microscope. Post PCR amplification, we need to check whether the promoter region of a highly expressed gene, like a housekeeping gene, has enriched during the process, while the gene desert region are not enriched. And also we can check the fragment size of the library by using the bioanalyzer or aggregate scale. We should see a laddering pattern, which is composed of fragments of different size, including the nucleosome-free region, the mono, di, tri-nucleosomer associated fragments. So if this is okay, so then we can use the high throughput sequencing to get the sequence. Then we use the fast QC or multi QC to check the raw reads quality and then mapping the reads to reference genome. Post the alignment, we also need using the ATAC-seq QC package to check the alignment quality and also the in silico checking the library quality. So before we use the ATAC-seq QC to check the library quality, we should do some pre-process. So first, we should check the quality, as I said, check the quality of our raw reads using the fast QC. And if there are multiple samples, you may use the multi-QC to generate a summary report on the HTML file. So if the fast QC reveal the many low quality bases or high contents of sequencing adapters, we may use the chemomatic or counter adapter to remove the low quality bases and the sequencing adapters. Then map the reads to the reference genome using the bowtie 2 or BWAMEM. So after that, the alignment files can be converted to the BAM file and sorted. So it also necessary to remove the contamination and the duplicates and the mapping artifacts. To remove the reads from the mitochondria or the chloroplasts, we can use the SAM tools. To remove or mark the duplicates, we can use the pickup tools. So to remove the mapping artifacts, so basically which including the fragments which are the less than 38 bases or greater than 2,000 bases, and the reads that are mapped uncoordinated and with a mapping quality lower than 20, using some customer scripts. After that, we can use the R package called ATAC-seq QC to do the comprehensive post-alignment QC. So the first function we used to use called BAM QC, which can be used to get the mapping status, such as the mapping rates, duplicate rates, genome-wide read distribution, mapping quality, and the contamination. And this the input for this function is a sorted BAM file with its index file also, with the duplicate reads mapped. Since the R implementation of this function is kind of slow, so you may also use the alternatives such as SAM tools or pickup tool to do this step. As I said, the TN5-mediated tagmentation process typically produce fragments with different size, such as a small fragment less than 100 base, which is from the nucleosome-free region, and other fragments that associate a single nucleosome or dinucleosome or even trinucleosome or tetranucleosome. So after the alignment, we can using the ATAC-seq QC package, there's a function called a fragment size distribution to get the insert size of the library. So on the left side showing a size distribution for a high quality ATAC-seq library. So from here you can see many reads are from the nucleosome-free region, which has a small fragment size less than 100 base. Then you can see a mononucleosome associated fragments and the dinucleosome, trinucleosome, even tetranucleosome associated fragments. On the right, showing the size distribution for poor quality ATAC-seq library. So basically, you can barely see fragments greater than 200 bases. So this probably is due to over-transposition of the chromatin. So the TM5 transposes cut the DNA, produce a line base overhang at the 5' ends. So to center the cleavage events, so we need to shift the aligned reads by the coordinates of align the reads. For those mapped to the positive strand, we shift the coordinates forward by 5 bases. For those reads mapping to the negative strand, we shift the coordinates by -4. So the function we can use is called shifted GA, shifted G alignment list. So next, we need to split the bound file based on the fragment size into different bins, so the nucleosome-free bin, mononucleosome, dinucleosome, and trinucleosome bins. Those bins, the reads in different bin can be used to visualize the signal later, such as we know that the promoter, the enhancer, and the insulator, those are in open chromatin region. So there are lots of reads from the nucleosome-free region should be visible in these regions. So in those open chromatin region, they are flanked by well-positioned nucleosomes. So you should also see the mononucleosome associated signal around the open chromatin region. So in the package, we have 3 functions, which call enriched fragments, which calculate the signal around the TSS using the split bound objects. And another function called featured aligned heat map, which can generate a heat map showing signal distribution around the TSS. And the mat plot can generate a density plot showing the signal around the TSS sites. So shown here on the left is a heat map, which has showing the signal from the nucleosome-free reads and the mononucleosome reads. So the red signal, so you can see there's a concentrated, they are concentrated around the TSS transcriptional starting site, while the mononucleosome signal, they have some pattern that flanked the nucleosome TNS transcriptional starting sites. On the right, showing the heat map of the signal of the nucleosome-free reads and the mononucleosome reads. So from poor quality ATAC-seq data. So from here, you can see the nucleosome-free reads, the small fragments are spread over a large region. So this is showing the over-transposition due to during the tagmentation process. To quantitatively showing the signal, we can use the mat plot to showing the enrichment of a signal around the TSS site from the nucleosome-free reads and the signal from the mononucleosome associated reads. So from here, you can see the nucleosome distribution around the TSS sites. While for poor quality ATAC-seq library, you can barely see the nucleosome distribution around the TSS site, even though you can see a widespread signal of the nucleosome-free reads. So after showing the transcriptional starting sites, visualize the signal around the transcriptional starting sites, we may also need to look at a broad region across the genome, because besides the promoter region, there are a lot of other regions like the enhancer, insulator. They also should be have an open chromatin. So we can use the genome browser, such as IGV, to realize many different genomic regions, including region with highly expressed gene and the region devoid of genes, such as the gene desert region. So in the package, we have the IGV snapshot function, which help to streamline the IGV snapshot process, which make the process pretty easier. So the transcription factor binding to the open chromatin region can interfere with the TM5 transposition process, which leave some footprint. So especially for the actively bound transcription factor, such as CTCF, so we can use the factor footprints to visualize the footprint. So for high quality ATAC-seq data, you should see a deep valley of the footprint. It's pretty deep, a deep footprint. But for poor quality ATAC-seq data, the footprint may be very shallow or even barely seeable. So once we see the library has a pretty good quality, we may ask whether we have sequenced the library deep enough. In the package, we have a function called a saturation plot. So basically, this function based on downsampling of the sequencing data and call the peaks using those downsampled reads, and count the number of peaks from each downsampled files. Then we plot the curve to see whether the curve flat out as the sequencing going deeper and deeper. So if the library was is not sequenced saturatedly, so we may find some library which has a good quality and a good complexity for further sequencing. So we can use the estimate library complexity function in the package to estimate the library quality, library complexity. Showing here on the right is the library complexity for different biological replicates. So for like this one, the purple line showing ATAC-seq assay with 500 cells, which has a pretty poor library complexity. So after sequencing like a 10 million, something like that, so the library was saturated. But for some other library, like this one, the what kind of green? So you can see the library is barely saturated, even after sequencing for 400 million. So if you just tell by the library complexity, the library complexity itself cannot tell whether the library is good or enough. So you should based on the other quality. So if the library has passed the other quality check I mentioned ago, then the high complexity library are good library for further sequencing. So now if we have an experiment which has assay from different conditions, multiple biological replicates for each condition, so we may have a bird view about the whole experiment by checking the reproducibility and also the consistency. So we have a function called plot correlation. So we can calculate the correlation based on the signal from each replicate. We can tell, so like the replicate 1, which is very different from the other. Replicate 4 is very, sorry, I mislabeled this. The replicate 4 is very different from the other replicates. So this function can also generate a principal component plot. To conclude this part, so I give some recommendation for current best practice for ATAC-seq assay and ATAC-seq data analysis. So to reduce the background noisy, so we may either digest away the background DNA from the median or the data cells using DNS1. So it's also important to prepare the nuclei from the fresh tissue or cryopreserved tissue cells. So the Omni ATAC protocol is one of the best protocol which can be used to reduce the mitochondrial or chloroplast DNA contamination. So if you use the standard ATAC-seq protocol, usually you can have up to 70% of reads that come from the mitochondria, so which is not quite worthy to do that sequencing, because most of the reads are not useful for your downstream analysis. It's also very important to optimize the ratio of the TM5 transposase and the number of nuclei to control the library complexity. So you need a limited the PCR amplification cycles. So qPCR should be used to optimize the number of cycle. Given the size distribution of the ATAC-seq library, so we suggest to perform the paired end sequencing. So we have a better estimate of a fragment size, and the read length can be from 40 base to 100 base. We see a lot of people do like 150 base paired end sequencing. So that kind of library is not quite necessary, because many of the nucleosome-free fragments are pretty small. It's around 38 base to 100 base. So you can have almost many reads that have adapter at the end, at the 3' ends. So depends on the purpose of the experiment. So you may sequence the library to 50 million reads, paired end reads for a general analysis. But if you are interested in footprint analysis, you may need to sequence more, like 200 million reads. As I said a moment ago, so after the sequencing, we may check the raw reads quality using the fast QC and the multi-QC. Then after the alignment, we need to use the ATAC-seq QC to perform comprehensive quality control to find out the best ATAC-seq data. To call in the peak for the open chromatin region, so we recommend a few peak callers, such as a gene reach, the max2, which are using the shift and extend options to call lateral peaks. More recently, there is a ATAC-seq physical peak caller called HHM-R ATAC, which require lots of computing resources like a RAM. So it's kind of slow. So if you have a good cluster, you may try this one. So after peak calling, so you may do further quality check, like using the chip Picano to associate the peaks to potential targets gene and generate a peak distribution among genomic features. And also necessary is that to do functional annotation of the peak to see whether the open chromatin you identified makes sense or not. So there is a tool, it's called a GRATE, it's a genomic region enrichment annotation tool, or a chip enrich, which can be used to annotate the peaks. In the last year's bioRxiv, there's a tool called BEHST, which can integrate the long-range interaction data to associate the enhancer, a distal sister regulatory like enhancer to a target gene. So if you are interested in the nucleosome positioning analysis, you may use the nuclear ATAC to find out the footprinting. You can use the tobias. To infer the transcriptional regulatory network, you can use the PECA2, but that tool requires you have a paired RNA-seq data and ATAC-seq data. So after that, I'd like to have some live demonstration. So in the demonstration, we have data from 2 different experiments. One is from the Greenleaf lab, which is, has a pretty high quality data. Another one is unpublished data, which has a pretty poor quality. So those I have done the raw reads QC and align the reads to the HG38. So the BAM file, the full set of BAM files are available from the Google Drive, but the subset, the filtered BAM file, are contained in the ATAC-seq QC workshop package. So we may use that. So the task gonna have, we are going to work through is first one is to check the read alignment statistics using the function BAM QC. Then we do a library complexity estimate. After that, we're gonna plot the library installed size distribution. And then we gonna plot the TSS enrichment, signal enrichment. And at the last, we're gonna use the IGV snapshot to visualize a few genomic region. So I'm not sure whether you have downloaded the Docker image for this workshop. If you have downloaded the Docker image, so you may find you can run the Docker image by do like this. So docker run -e setting a password, then starting the set a short name, then the Docker image name. After starting that, so you can find the R script for the live demo in the ATAC-seq QC workshop package in this folder. So first, you can load in the package. So after loading those packages, what I did, then we can read the BAM file, which is with duplicate duplicates marked. Then we can run the BAM QC function to check the mapping statistics. So this function is kind of slow. It may take a while to finish, even though we only use the chromosome 20 alignment file here.
Yes, now is sharing the
Yes, now it works.
correct screen. I'm sorry for that. Just we already run the BAM QC, right? It's just talk you the basic information about your BAM file. I feel it is too tiny of the font.
Yes,
Okay.
if you zoom it up, yeah. Mm-hmm.
We just next step is we check library complexity. It's called estimate library complexity, and you can see the library complexity is something when the plot is like this. The x-axis is a subsample of the fragments. The y-axis is a unique, distinct fragments. And then we can what's this plot library insert section. For this part is that only paired end can use this function because we can get the fragment size by the paired end sequence. But for single end, to calculate the cross correlation between positive and negative strand reads maybe a one method, but it's still time-consuming work for the single-end reads, and the calculation may be not as correct method to estimate the fragment size for mononucleosome or nucleosome-free or dinucleosome region. So we are still thinking how to apply this function to the single nucleotide, single end sequence. And the part 3, when we run the, this is just give the label for your figure. When we run it, it will generate the fragment length distribution. You can see this is just as I have already told you, this is a nucleosome free reads mono. Oh, this is mononucleosome, dinucleosome, trinucleosome. If we show it in log scale, it will show more clearly about the distribution. This part, we just try to guess tags in your BAM file. Indeed, this part, if you already know, you can you can view your BAM file by some tools to know the tags. What should you import in the BAM in the new BAM file? We include this part just want to include as more as possible the information for the output BAM file to you. You can sometimes you will get an error when you use some text that is not involved in the all reads, so you can just remove that text. We set outputs to save the splitted BAM. This one is just in case you already tried this script. And then the seq level is that because this sample file is only contain chromosome 20. Lot of people complain that the running time for ATAC-seq QC is too long. We are continuously trying to reduce memory cost and running time, but the thing is that we still have trouble in increase the efficiency of the code. Anyway, I think current version is in a acceptable memory cost and running time. In the future, we will still trying to improve the efficiency of the program. Object is not fun. SIG level, I did not run this step. Okay, this read BAM file, if we set the big file as true, then indeed it will not read the BAM file into the memory. You can see it will give you 0 elements. Indeed, it will read the file later, because if we read the file load all the BAM file into memory, it will cost a lot of memory. So I designed it to read it later. And after that, we can run shift J alignments list. This one, we already also have a different version of this function for the single end ATAC-seq. So you can also use shift alignments, a different function for the single end reads. This one is used for the paired end. It will cost a little time for running shift alignments. Basically, it will shift the reads for positive string for 5 nucleotide, 5 nucleotide and negative string manners for nucleotide. Basically, it will fill the 9 nucleotide by gap. After that is that we after that we can this is basically some information for the transcriptome. Previously we are using this one to assign the possible mononucleism, trinucleism, or binucleism reads, but currently it's just double check about the sequence name. If you do not fill up the conservation score for that, so this step is used to split G alignments by the sequence by the fragment size. It will output the fragment into mononucleism or dinucleism or trinucleism, depending on the fragment size. After that, we can check about the output file. It will contain a lot of information. So you can see the split BAM file folder contain a lot of them. For example, the dinucleism BAM, it will contain the dinucleism file, the mononucleism file, nucleism free file. All this one can be used for downstream analysis. Also, some of the reads, the read length is between the, for example, nucleism free and the mononucleism. Okay. Then we can do some we can do some metagene analysis. For example, we can plot the fragment distribution like this. This is just subset. So you can see in the nucleism free reads the heat map for that one. And this one is just want to repeat the figure in the green leaf lab. They plot the nucleism free reads and see the peak and of upstream and downstream of the transcription start side. You can see the the distribution of mononucleism, dinucleism, trinucleism. And also, you can also, oh, this one asks ask the IGV installed and the port open. So in the Docker, it is not installed, so we cannot do that one in the Docker. Okay, give back to Haibo.
All right, well, thank you, Zhanghong. It's great you rescued the session, and we are pretty much at the end of it. We have lots of questions, and questions in polls, they are anonymous. If someone who would like to get answers to those questions, please type your name and question in the chat window, and I'm pretty sure Haibo will follow up and answer those questions. And that concludes this session. Thank you all for coming, and that's it.